In this video, we will be discussing about transcription, which is the process of making messenger RNA, or in short, mRNA. First of all, it is known that an mRNA molecule is made by copying or transcribing a gene sequence in a cell's DNA, which is found in the nucleus. In the nucleus, we will find DNA double helix. By looking close up, we see that this DNA contains genes, which is a continuous ring of nucleotides containing a region that codes for the mRNA molecule. This region begins with a promoter and ends with a terminator. In detail, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. After that, the majority of gene expression is controlled by either permitting or blocking access to this site by the RNA polymerase. Further close-up on the binding site, the DNA double helix unwinds and opens. This region of open up DNA is called a transcription bubble. Next, the RNA polymerase starts using one of the DNA strands as a template to make a new mRNA molecule in the 5' to 3' direction by pairing up complementary bases. In that case, the RNA product is complementary to the template strand and is almost identical to the other DNA strand, called the non-template or coding strand. However, notice that all of the T nucleotides are replaced with U nucleotides in the newly made RNA. In the meantime, RNA polymerase slides along the template DNA strand until it reaches the terminator region. At that point, the mRNA transcript, the RNA polymerase, and the DNA strand dissociate from each other. By then, the mRNA transcription is now completed.